to discuss the analytical solutions that Shimadzu can offer when it comes to vaccine testing. It is a great pleasure to introduce to you our guest speaker, who is none other than Liu Wantung. So prior joining Shimadzu, Wantung started her professional career as a QA/QC chemist in a multinational pharmaceutical company for 3 years. She was formerly our senior application chemist in Asia Pacific Singapore, and currently, as the LC product manager, she provides technical and marketing support on Shimadzu LC and LCMS products, as well as for pharma market across Asia Pacific region, including Philippines. Moreover, she specializes in method development of biopharma and pharma sample matrices. So without further ado, let's all welcome our key speaker for this webinar, Wantung. Wantung, please.
Hello, greetings from Singapore. As you can see, my background is Marina Bay Sands. Yeah. Hi, this is Wantung from Singapore, Shimazu, Asia Pacific. Thank you, Laika. Okay, and it is a great honor to meet all our Philippines friends on this live webinar. Okay, so without further ado, so today my topic that I'm going to share with you with all our online audience here, we have 76 participants. I can see from the screen here. So, okay, let me start sharing my screen now. Okay, so this is my topic for today. This is what I'm going to share with you is the analytical intelligence, the AI and pandemic. Okay, so first of all, right, I believe all of you agree with me that okay, hold on, let me. Sorry, for a better. Okay, so I believe all of you agree with me that we are very much affected by the pandemic right now. Okay, let me share my screen again. Okay, so can you see my screen, Laika?
Yes, Wantung, we can see it.
Okay, awesome. Okay, so as you all know that we are now, the world is struck by the COVID-19 pandemic, and many of us are being affected, and it is completely a new thing that we are allowed to work from home. Last time, right, in the past years, we have never thought of working from home. But now, because of this pandemic, we are forced to work from home now. So as you can see on my screens here, right, this is not said by me. This is like from the United Nations. The United Nations urged the government to need to make full use of the digital technologies to confront the COVID-19 pandemic. Okay, so this is what we have understand from the United Nations that, okay, we have to use the digital technologies. Okay, so digitalization of the laboratory workspace has become more urgent and critical than ever before. Okay, so as you can see, the digital technology has been growing rapidly. So I am sure that you agree with me, people start to require more assistance from the digital technology. Okay, so as you can see, right, we need the more assistance from the digital technology. Okay, so I will be sharing on how our Shimadzu new Nexera series, right, that incorporated with the artificial intelligence, and we call it the analytical intelligence, okay, the Internet of Things, machine learning, okay, so reduce the labor work and release our user from the routine instrument operation. Okay, so maybe Laika can help me to share the first poll question before I continue with my sharing. Okay, so yeah, Laika, please.
Great. So here is our first poll question. Actually, we have 3 poll questions for this session. So here is the first one. The question is, are you currently using Shimadzu LC system? So just click or just select yes if you are a user of any Shimadzu LC system, and click no if not yet or not. Okay, so we are seeing more than 50% of the attendees has responded already.
Okay.
So this is a good, we have a good sort of attendees that they are, yeah.
Yes,
Okay.
That's good.
Okay, so last 5 seconds for you to select your answer. So again, are you currently using Shimadzu as a system? Select yes if yes, and select no if no. So last 2 seconds. All right. Okay, so now I'll be sharing the results. So 17% are users already. 17% of our attendees is a user already of Shimadzu LC system, and then 83% are not. So back to you, Wantung.
Okay, thank you, Laika. So that's the reason why, okay, today is the best time for us to understand the Shimadzu AI function with our new Nexera series better. Okay, so thank you for all the audience participation in this poll question. All right, so now let's continue with, as you can see here, okay, so basically for the Shimadzu Nexera series, right, as you can see, it is a new benchmark of intelligence, efficiency, and design. And we have the 3 models, which are the Nexera XR. As you can see here, the Nexera XR, X3, and the XS. So basically, right, 3 different models, they have 3 different pressure tolerance from 70 to 130 megapascal. And all the models here, they have the AI functions. Okay. So I will share from the intelligence, the benchmark of intelligence first. As you can see, I believe all the chemists here, you agree with me that this is our routine in a lab, our daily routine in a lab, right? So as you can see, most of the steps here, it requires the chemist to stay beside the instrument, okay, most of the time. Okay, so however, with the Nexera LC40 series, right, the whole process can be done automatically from the startup, okay, auto startup to auto shutdown, and mostly can be done unattended and eliminate the risk of failure. So today I'm going to share mostly on these 3 unique features of the Nexera LC40 series, which are the mobile phase monitoring function, the flow pilot, and also the auto recovery function. Okay, so I will proudly say that Shimadzu, we are the first and only company that have the gravimetric sensor. Okay, as you can see here on top, this is the gravimetric sensor that is on top of our LC to constantly measure the real-time volume of the mobile phase. And this gravimetric sensor, it will alert you when your mobile phase is insufficient. Okay, so we are human after all, and human, we do make mistakes. Sometimes when we run a very long batch sequence, right, we might miscalculate the required mobile phase volume for the batch analysis, and the amount of mobile phase that we prepare is not enough. So after your batch sequence is registered, right, this window, it will pop out. Okay, this window, as you can see on my screen now, it will pop out, and then it will show the total mobile phase volume that is required for your batch analysis. Okay, so as you can see, right, the first column, it will show the mobile phase volume used for the current batch. If you have few batches in the queue, the second column is going to show you the total mobile phase volume that is required. And then it will also show the remaining volume, okay, that is in the bottle. Okay, so if let's say your mobile phase that you prepared, right, is not sufficient for the entire batch sequence, the window here, it will show you that the mobile phase is only sufficient to analyze up to 11 lines in your batch sequence. So this system is so smart that it will automatically calculate the mobile phase usage that you need for you. Okay. And then as you can see here, okay, this is the example after the batch sequence is registered. Then you can see here, this is the window that show the total usage volume and the remaining volume that you have in your mobile phase bottles. Okay, so for the mobile phase monitoring activation, right, it can be done on the lab solutions, which is our LC software. Okay, and now this is the mobile phase monitoring setup is also very simple. As you can see, it is just as simple as 3 steps. Okay, so this is just like, I'm sure most of you here, you use balance before. So when you want to calibrate this gravimetric sensor on top of your LC system, right, it is just like calibrating a balance, just to tear the device here and then to calibrate it with your mobile phase. Okay, so you can also set the warning and the error volumes. Okay, for example, if let's say you want the system to alert you that your mobile phase is below 300 ml, right, you can set accordingly. So 300 ml is the default warning volume and 100 ml is the default error volume. So you can also download the mobile phase monitoring app in your smartphone so that you can monitor your mobile phase and your analysis anytime, anywhere. Okay, so this is how the AI feature of the system is assisting you to help you to reduce the time that you need to spend in your lab. Okay, so the system has the auto startup function as well. So you can actually program your system to start the run even before you reach the lab. For example, if let's say in Shimadzu, we start our work at 8:30, but before that, I want to purge and condition my system. I can let the system automatically wake up at 8 o'clock in the morning, even before I reach my lab. So by the time when I reach the lab, the system is already ready for me to put in my sample and standards to run the system immediately. Okay, so this is the auto startup function. Okay, and then the next I'm going to share with you is the conditioning of the column and the system. Okay, how to do the conditioning of the column with the flow pilot function. Okay, so the flow pilot function, right, is to maintain the column lifetime. I'm sure all of you agree with me that column can be very costly also, right? So if you can prolong your column lifetime, right, you are helping your lab to save the money. Okay, so this flow pilot, it is necessary. Okay, so it is always necessary to increase the flow rate gradually while observing the oven temperature. So as a chemist, we all know that if let's say you increase your flow rate immediately straight to 1 ml per minute, right, you are going to give a pressure jump to your system and your column. And this scenario is going to damage your column because we are not supposed to give a sudden pressure shock to the column, to our delicate column. So we have this function called the flow pilot that it works in conjunction with the oven temperature, meaning before your oven reach the set temperature, for example, 40 degrees Celsius in this case, before the oven temperature reach the set temperature, right, the flow rate, it will gradually increase up to the half of the set flow rate. In this case, half of the set flow rate is 0.5 ml per minute. Okay, so it will stop here and wait for the column oven to reach the set temperature. Then only it increase another 50% of the set flow rate up to 1 ml per minute. Okay, so this function, right, is to protect the column so that to extend the lifespan of our delicate Hblc column. And the main purpose is to maximize the return from our cost of operation. Okay, so now I would like to show a very quick video on how this function works. As you can see on the screen now, it is after purging the pump. Okay, so for example, if let's say in this case, my flow rate is 1 mil per minute, right? In total for A and B, you can see the F here is showing that the flow rate is increased up to the half of the set flow rate. Until the column oven temperature reach set temperature, then it increase another half. So the green light shows that the pump is ready for your analysis now. Okay, so this is the flow pilot function, which is the second AI function of the Nexera LC40 series. Okay, so now, okay, this is the flow pilot setting in the lab solution software. So I'm proud to say that we are the first company, right, that have our flow ramping function works in conjunction with the column oven temperature. Okay, so the flow pilot setting, okay, if you want to understand this function more, right, okay, you can always raise your question, or you can always look for our SPC chemists, and they will be glad to explain this to you. Okay, so the next function that I want to share with you is the auto recovery function. Again, the auto recovery function, right, it is Shimadzu patented self-monitoring and corrective mechanism. Okay, so how does this function work? In an extremely rare case, when you have an air bubble, okay, for example, this is the normal pressure. Okay, so for example, if let's say you run a long batch sequence overnight, and then you have an air bubble that enter your Hblc system and impact your batch analysis, right? Imagine it is in the middle of the night and you are sleeping, nobody is in the lab. And if this scenario happen, right, okay, usually it will cause the pressure to drop, and then it will affect the whole batch of analysis. Okay, but this auto recovery function, right, it helps you to automatically diagnose and detect this scenario so that the system will very smart to repurge your system. It will do an auto purging until the pressure is back to the normal pressure, then continue with the analysis. So your batch analysis, right, will not be affected by this air bubble that coming from nowhere. Okay, so you can see this is how it works. Okay, for example, this is the video that I want to show you. Imagine in the middle of the night when you run a long batch sequence of analysis, then you have this air bubble that enter your system, right? Okay, as you can see here, there is an air bubble in the red and circle in red here. It enter your system. Okay, so what happened is that your analysis is running. Okay, so this is supposed to be the normal pressure that you should be expecting. Okay, the line here. So there is a sudden drop in the pressure due to the air bubble enter the system. Okay, then the system, right, will detect this kind of abnormality, and then it will quickly do a purging to the system. As you can see now, okay, the pump has purged and then resume running. The pump has started now. Okay, so when the pump starting, right, the flow pilot function is also initiated. Okay, so now you can see that the pump pressure is back to normal again. Okay, so this is how the auto recovery function help you when you are not in the lab. It help you to recover your system. Okay, so this is the third novel feature that I want to share with you. Okay, so this is the auto recovery setting that you can do in your lab solution software. Okay, and then for example, right, if let's say your batch analysis is being affected, okay, the system will re-inject the affected analysis, and then it can also do a blank injection to help you do the column cleaning. Then after that, followed by the re-injection to acquire the injection of the sample that was affected just now. Okay, so for those customers that are working in the regulated labs, like for example, those pharma labs, right? If you are working in a pharma labs, okay, so now your concern will be, is all this being captured? Okay, so this, everything will be captured in the log browser that shows what happened to your system when you are not around. So everything will be captured here in the log browser. Okay, so this will be the log file in the auto recovery. So every single thing happened, right? What time it happened, at what time, and then what has the system done, everything will be captured here in the log file. Okay, so if you are working in a regulated lab, right, you do not have to worry about that because everything will be, it will be recorded. Okay, when the interrupted analysis happened and then the pump purged to remove the air bubbles, everything is recorded. Okay, okay, so far I have already explained all the 3 unique and novel features of our lab solution, of our LC40 series. Okay, which are the first, mobile phase monitoring, second, the flow pilot, and the third is the auto recovery function. Okay, so these are the 3 AI features. Okay, so now you can see that in summary, right? When you arrive at the lab in the morning, right, with the auto startup function, the system has already is in the ready status. Okay, so what you need to do is just do the column conditioning, and the column conditioning, right, with the flow pilot function, it behaves as if the system is handled by a very experienced chemist. So before you start the analysis, right, with the mobile phase monitoring, the system will tell you whether your mobile phase is it sufficient for your entire batch of analysis. Okay, so if anything happened during the analysis, right, the auto recovery function will also help you to detect and then recover the system by itself. So overall, right, with these intelligent functions, basically you can spend less time with your system in the lab because the system is so smart that it can just auto monitor and recover by itself. Okay, so especially in this era, right, when we are all in the pandemic, right, if you need to work from home, you can still monitor your analysis from your home, okay? So next, we will move on to the Nexera LC40 new benchmark of efficiency. Okay, so we noticed that there is a growing demand for high analysis throughput, especially in the pharmaceutical and the clinical laboratories, okay, to increase the lab productivity. So the auto sampler SIL40 series, it can achieve ultra high analysis throughput with the fastest injection speed. Okay, as you can see on my screen now, the injection speed of the auto sampler is only 6.7 seconds. Okay, and 8 injections of caffeine can be completed within 1 minute. Okay, and as you can see here on the screen, this is the plate changes, which you can connect a few plate changes and it can help to increase to achieve ultra high sample capacity. Okay, so maximum sample capacity can be up to 16,000 samples if you want to really increase your lab analysis throughput. Okay, and then the second thing is the ultra low carryover. Shimadzu auto sampler is very well known for ultra low carryover. So as you can see here, the carryover after the caffeine injection, right? If it were to detect by MS detection, the carryover is only 0.0002%. So it is very, very low, and it is the lowest in the market so far. So zero volume connection between the sample loop and the needle ensure no sample residue. So ideally designed to achieve the lowest carryover. Okay, so the next one that I want to share with you is the air circulation temperature control. As you can see here, the new design of our auto sampler helps to block the air from outside. Okay, so this kind of new design, it will effectively prevent dew condensation. Okay, the auto sampler is delicately designed to consider airflow direction within the sample compartment to make sure that stable temperature and effective cooling. So this is the new design to improve the auto sampler, this auto sampler compartment. Okay, so the last but not least function that I want to share with you on the auto sampler is the automated preparation function. So the auto sampler is also designed for automated dilution, reagent addition, and also you can do some, for example, if let's say you want to do stack injections, it also can be done with the auto sampler. Okay, so this is also a patented technology, okay, to do the automated dilution and everything. So as we know that dilution, right, usually it is done by the chemist, okay, but if you have this function in your auto sampler, right, you just need to program the software and then the system will help you with the dilution automatically. Okay, so the dual injection mode here, right, for example, if let's say you want to do two types of analysis, as you can see on my screen now, for example, some of our users, right, they want to run a single sample with two types of different analysis. For example, I want to run amino acids and the organic acids, right? So I can do it in the dual injection system. So I just put one sample in my auto sampler, and then with the two different pathway, I can inject the same sample but go through two different flow paths to acquire two different results for my analysis. So this is the dual injection mode. Okay, this integrated two analytical system in a single platform enables the analysis of two independent samples through the two different flow channels to acquire the amount of sample processed in the same time. Okay, so now let's move on to the new benchmark of design. Okay, so here are the new features of our PDA detector. Okay, so as you can see, this PDA detector, I believe for those participants that you are using the LC, right? I'm sure most of you are using either the UV detector or the PDA detector. Okay, because most of the compounds, right, they have the conjugated double bonds. They can be detected by the UV or PDA detector. Okay, so now, as you can see here, this is the new feature of the PDA detector. With the advanced TC optics technology that control the temperature of the lamp, it helps to eliminate the influence of the room temperature drift. So as you can see on the screen, with the new PDA detector, right, the baseline of your chromatogram will become very stable compared to the other vendors' PDA detector, whereby they have a drifted baseline. Okay, so this is the new design, new technology in the PDA detector. Okay, as you can see here, the dynamic range of the detector has also been improved to 2.5 AU. So this enables simultaneous determination of low and high concentration samples. Okay, for example, if let's say you are doing some API and the impurities analysis in your pharma sample, usually the active pharma ingredients is high in intensity, but the impurities are small in terms of the intensity. They are relatively low in intensity. So with this kind of wide dynamic range, right, you are able to detect both high concentration and low concentration compounds in your sample. Okay, so this is the excellent dynamic range linearity. And as you can see, this is the futuristic, sleek design of the Nexera series that promises no compromise on the performance. Okay, so the previous system is wider, but now the new Nexera LC40 system, right, is more compact. Okay, so in terms of the space saving, it achieved 40% of space saving. In terms of the power saving, when your system is not running, you can let the system in the standby mode. So it helps you to save 68% of the power. Okay, so, okay, now the hot topic is, right, usually we are saying that we want to work from anywhere, right? Okay, I'm not sure in Philippines, but in Singapore, our government, right, is trying to urge like 50% of the manpower should be working from home, okay, because of the COVID-19. So now the question comes to, how do we work from anywhere, especially like working from home with compliance? Okay, so this is how you can achieve by using the remote operation and monitoring using the lab solution CS. Okay, for example, if imagine you are the user, you can access your lab solutions and your system anywhere, even when you are working from home. Okay, so all operations can be performed, including the instrument startup, analysis start, monitoring, and shutdown. Everything can be done from home. Okay, so this is the usual workflow. Okay, all you need to do is just to do the preparation of the standard and the sample. Okay, after you set all the vials into the LC, right, and you do the column installation, basically you just need to spend maybe like half day in the lab. The rest of the thing like running sequence, monitoring, data processing, everything, check progress, all this, you can do it from home. The system is also designed for like automatically shut down. So after the batch analysis completed, right, the rest of the things that you can actually do from home. Okay, so this is how you can work in the comfort of your own home. Okay, so remote operation is very essential. And also for those who are working in the pharma QA/QC lab, right? I was also from the pharma QA/QC lab. So we know that the 21 CFR Part 11 is very important, the compliance. So the lab solution CS, right, it meets all the parameters and the criteria here to be 100% fully compliant. For example, like the validation, the human readable output, the audit trails, version controls, in electronic signatures, backup and restore, everything can be done with our lab solution CS. Okay, and for you to know, these are all the instruments that can be connected under the lab solution CS network structure. Okay, from LC to GC to lab solutions, LCMS, okay, and even our other vendor system can be connected under the lab solution CS as well. Okay, so now we move on to some of the applications that I want to share with you. Our new research application notes that has been published. Okay, so I'm sure now the hot topic is about our new COVID-19 drugs that are being approved by the FDA. Okay, as you can see, right, some of the drugs, right, it has been approved by the FDA as the first treatment for the COVID-19, including this compound called the remdesivir. So if you are interested in this application, right, you can always ask from our SPC colleagues, and they are more than happy to share with you. So we have Shimadzu, we have already published the application on the remdesivir. This is the first treatment that is approved by the FDA for the COVID-19 treatment. Okay, so if you want to understand more, please contact our SPC colleague and they will help you. So the COVID-19 treatment, right, the other one that FDA has approved for the COVID-19 prevention, right, in the US nursing home is this drug called favipiravir. So favipiravir, we also have an application, okay, that we did it, the quantitative analysis of favipiravir in the plasma by using the Hblc. So if you are interested in how the application can be done on our system, right? Always contact our local colleagues and they will help you, okay? They will share this with you. Okay, so the third one, right? US FDA has approved the first 2 oral anti-COVID treatments. One is the Pfizer Paxlovid and the other one is the Merck's Molnupiravir. Okay, so this is the methods that we have. So it is a simple and it can be done on the Hblc. So please contact the local colleagues to help you with all these relevant applications. Okay, Shimazu, we are always be there to support you. So if let's say the other applications that you are interested, right? We have a lot of applications that is in our website. Always contact our local support team and then we are here to support you, okay? Even from Singapore, I can also, we work together to support our customers in Philippines, okay? So this is my first presentation, okay? So before we move on to the second presentation, right? I need Laika's help to share the second poll questions. Okay.
All right. So here is the second poll question, 1, 2, everyone. Do you need to do sample purification in your analysis workflow? So may it be any application, may it be any analysis. So just answer yes. If do you need sample purification step in your analysis, and no if not. Okay. So far we have already 31% of the attendees who participated, and we can give you another 5 seconds to answer this poll question. So again, do you need to do sample purification in your analysis workflow? Select yes if yes and no if not. All right, so last 2 seconds. 1, 0. So I'll be ending the poll and here's the result. So 1, 2, everyone. 74% of the 57 respondents answered yes, they need sample purification in their analysis workflow. And then 26% answered no. So back to you, 1, 2.
Okay, thank you, Laika. Okay, I'm a bit surprised to see this kind of ratio. Okay, frankly speaking, I didn't know that our users in the Philippines, you all need to do sample purification that much. Okay, so luckily I prepared this specially for you guys. Okay, all right, so now, okay, allow me to share the second presentation for this webinar today. Okay, it's about the sample purification. Okay, sample purification is in terms of, for example, like you want to, for example, from analytical LC, then you want to scale up to prep LC, and then you do the purification of your compound. Okay, so now I off my video first, and then I share my slide. Okay. Okay, so wow, we have 103 participants together with me on this live webinar now. Okay, so it's my pleasure to share with you the overview of Shimadzu solutions for the purification workflow. Okay, can you see my screen?
Yes, okay. We can see it already.
Okay, awesome. Thank you, Laika. Okay, so purification workflow, right? Okay, what does it mean? Okay, this is the overview of purification workflow. For example, right, usually we start from the extraction to have the mixture of the sample. Okay, after you extracted your target compound, Okay, it could be by liquid extraction or solid liquid extraction or some other methods that you do to extract your target compound. Okay, after that, you need to do method development, especially in the R&D lab. You need to decide the conditions for your analysis, choosing the right column, choosing the correct mobile phase for your identification of the compound and achieve the separation. Okay, after that, right, of course, you need to estimate the goal, your target final amount for your compound. So in that case, you have to decide how much you need to purify in terms of the target amount. Okay, then only you decide how you want to scale up from analytical to the preparative. Okay, so if you want to understand how to choose the column size, right, you can always contact us. We will be there to help you also if you couldn't decide. Okay, and then after that is setting the prep parameters. Okay, so of course you need to get your compound separated, then only you do the fractionation. Okay, fractionation is what we call to purify the compound and fractionate the compound. Okay, so it's either you can do a reanalysis to confirm that the compound that you fractionate and you purify is your target compound. Okay, so this is the reanalysis. And alternatively, if your solvent contains some salt or buffer solvent, you can do a desalination, changing the solvent concentration, or you can dry the sample so that you can ultimately get your target compound, the yield, okay, in the powder form. Okay, so this is the whole process of the purification workflow. So I will introduce step by step. Okay, first of all, of course, the method development. And I believe all the chemists here, you agree with me that this is the most tedious step, okay? But not to worry, for the method development, right, we have the method scouting solution from Shimadzu that can help you. Okay, especially it is the most challenging part is how you need to decide the correct column, the appropriate column and the mobile phase for your analysis. Okay, so this is the Hblc, the normal UHBLC, the analytical scale. Okay, so first of all, right, if you do not know which mobile phase is the suitable one, you can use the method scouting solution to help you to scout the best combination of mobile phase. And also with this method scouting solution, right, you can scout automatically with different types of the separation mode using different types of the column. Okay, for example, in terms of the reverse phase column, we have many different types. We have C8, C18, C4, C1, or even some of the stationary phase, right? Like the phenol type, like the PFPP type. So if you are not sure which one is the best, right, you can scout automatically using this kind of setup, which we call the method scouting setup. Okay, so the software will help you to create different combinations. So you can try on it and then do the scouting, and then the chromatogram will come out, and then you choose which one is the best separation and based on what kind of the separation mode and what mobile phase. So this is to simplify your method development workflow. Okay, so after you have decided which column and what mobile phase combination, then only you scale up to the preparative mode. Okay, so here as you can see, when we say scale up, right, it is from the analytical scale, for example, like a conventional column. For example, usually the conventional analytical column is up to 5 mmID. So if you want to do purification, you scale up to the preparative scale, for example, like even up to 50 mmID for some of the prep columns. Okay, so this is an example of the scale up. Okay, and then after that, right, so with the Nexera prep system, right, one important thing is that multi-sample and multi-fractions, okay, can be the additional FRC, the fraction collector, can be connected to increase the fraction number and various allowable fraction containers. Okay, so this fraction collector, right, the LH40 system, right, it is a customer-oriented PrepLC supporting function. Okay, and it has like 35% decrease in terms of the installation width. In terms of the system width, right, it is like more compact. Okay, so the beauty of this system and software is that it has made your fractionation so easy. For example, if let's say I want to do fraction collection for this particular peak. Okay, the software is very user-friendly. You just need to take the peak start and the peak end, and then it will reflect the peak detection parameters on your software. And then the system will automatically do the fractionation for you. Okay, so this is how you auto create your fractionation parameter setting. Okay, this is the new feature. Okay, so for example, right, we are very familiar with this kind of the analytical mode. Okay, for example, you have 4 mobile phase running on an analytical system, the analytical kit. Okay, so then after that, right, when you scale up to the prep, you can just do a fractionation and do the collection in your fraction collector. Okay, so this is how the LH40 looks like. Okay, so the feature of the fraction collector and the liquid handler, right? What's the difference between them? Okay, I'm going to share with you now. So this is a fraction collector. As you can see on my screen here on the right side, it helps you to do the fractionation. For example, if you have 3 peaks separated by your column, so you can do the fractionation each peak into different test tubes. Okay, and then after your compound being detected by the detector, then the fraction collection into each and every test tube. Okay, so the fractionation happens when you set the parameters, and when your peak reach the threshold of the signal, then it will trigger the fraction collector to collect the fraction in the respective test tubes. Okay, and this is how it looks like. Okay, you can program it in such a way that for the baseline, you let it go to the waste. For your target compound, you switch to the fraction collector to do the fractionation. Okay, and then another improvement in the prep system is that last time only able to connect to one detector, okay, for the previous model. But now with the new fraction collector, we can connect to up to 4 detectors. For example, if let's say you have 2 detectors, okay, some of the compound can be detected by UV detector, some detected by RI, the refractive index. So you can trigger the fractionation accordingly, okay, based on the setting parameters. Okay, for example, these compounds, right, they are only detected by the UV detector. Okay, then you set the parameters, and then this compound, when the intensity reach the set value, then it will be fraction collected. Okay, another one is the RI detector, because we understand that some of the compound, right, like for example, some of the saccharides, sugar, alcohols, they are only detected by the RI, refractive index detector. So the fraction collector, right, you can connect to different detectors, so that different detectors trigger the fractionation, right, and it will collect accordingly. Okay, so this is the detection signal. Okay. Okay, so the liquid handler, right, you can see the difference between these 2 fraction collector. The FRC40 that you see on the right, it is for fractionation. When you want to collect your fractions you can collect it in the FRC40. And the liquid handler, right, what it can do for you is not only it can do the fractionation for you, but also you can see here, it can do the re-injection as well. Okay, the LH40, the liquid handler 40, it has the injection function in addition to the function of the FRC40. Because in some of the cases, right, most of the cases, customer want to re-inject the fractions that they have collected, okay, in order to confirm that the purification and the fractionation that they have done is the correct compound. So they need to do re-injection. So you can do that with our liquid handler 40. Okay, so LH40 have two main functions. It is being a role as a fraction collector and as well as an auto sampler. Okay, so one device, it can do dual job for you. Okay, so it can be, you can collect your target compound and at the same time can re-inject into your system to confirm. Okay, so this is the mixture that you have extracted. And then after that, you can inject and then do the fraction collection. For example, you want to collect these two peaks, fraction A and B, you can do it in the LH40 and then re-inject again to confirm. Okay, so re-injection. Okay, then you can get your analytical results. So it has a dual function in one single device. Okay, so, and most importantly, right, if some of my users, right, they tell me, hey, Wantong, because my compound is very special, I have no idea when the compound, or I want to do manual fractionation. I want to decide from where to where I want to do the fractionation. You can do it manually because we have the graphical user interface here. So it's very easy. By the time when you see your peak, right, you can open and close the fraction collector manually. Okay, we do have some customers, right, they want to do it manually because they do not want to miss any of their precious sample. So if you want to do it manually, you can do it on the lab solution screen here. If you want to do auto fractionation, right, it can be a simulation by lab solutions. You can simulate, for example, I want to fractionate this peak, okay? Then very easy. You just need to use your mouse cursor to pick the start point and the end point. And then the software, it will simulate the parameters that you can use to fractionate this compound. And then after that, you just click, and then the parameters will reflect. And then this successfully do fractionation for your compound. Okay, so it is as simple as that. Okay, so you have the choice of doing manual or you want to do it automatically, also can. Okay, so this is the summary of my sharing for this topic. First of all, right, if you want to do method development and if you want to understand our method scouting solution more, you can contact us and then we will be there to support you. And then you can scale up to the prep LC, and with the liquid handler, you can do the fractionation and do the re-injection. After that, do the post-treatment and dry out your system and get your amount of the target compound that you have purified. Okay, so thank you for your attention, and that's all for my sharing today. So now is our Q&A session, and I welcome our users and our participants. You can ask me any questions that you want to understand, or you can just ask me any question. Okay.
Yeah. All right. So thank you, Wantong, for the very informative presentation. So I'm pretty sure that all of us here have learned a lot, the Shimadzu solutions for the vaccine testing specifically. So I have receiving questions already in the chat box, but you can still type yours. Or if you want to talk to our speaker personally, so you can just type your name and company name in the chat box, so we will give the floor to you. But before that, let me just read the first question. Okay, so this one, can you also automate cleaning of the column?
Okay, thank you, Bernard, for this question. Okay, actually, right, it's very important because we have to clean our column from time to time, right? Usually, I will encourage my user to flush the column after each batch of analysis. So, Bernard, you can do that. Okay, basically, right, usually I will encourage my user to program and to set a column flushing method after your batch of analysis. So after the batch is completed, right, automatically it will enter the column flushing procedure. Okay, so you can automate that. Yeah, so my answer to Bernard is yes. Okay, if you want to understand more, please contact our local chemists and they will be glad to help you and guide you how to do that. Okay, so Bernard, yes.
All right. Okay. So for our next question, actually, this question was asked by one of our registrants. So it is stated in her registration. Okay. So can you list where is it? Yeah. Can we ask a list of possible equipment to be used for vaccine testing?
Okay. Okay, thank you. This is a very good question. Okay. So basically, right, most importantly for vaccine testing, right, LC cannot be escaped. So that's why today the main sharing is on the LC. Okay. And then for those compounds, right, they are very trace in amount. Okay. We need LCMS for the sensitivity. Okay. So LC and LCMS is more or less like it is a must in the list. And also some device, right? Like for example, the MALDI-TOF is also very important. But the thing is, right, not to worry because we have a solution of the vaccine testing. We have a total solution for the vaccine testing. Okay, so our colleagues, right, like Laika, like Faye, they will share the solution book with you. Okay, so it's an entire total solution for your vaccine testing. Okay, but LC is definitely a must. Okay, for the vaccine testing.
Yeah, so maybe after this session we can share to them our booklet on our Shimadzu solutions for the vaccine testing.
Yeah, it's a complete solution for our users.
Yeah, correct. Yeah. So a while ago, a number of attendees also messaged me to share with them the link of our application note on the analysis of favipiravir as well as for remdesivir. So guys, by the way, I have already typed the link in the chat box, so you may check on those. So those are 3 application notes. So moving forward to the next question. So can you do auto fractionation right as the chromatogram is generated? So can the Hblc do the automatic fractionation as the chromatogram is generated? So 1, 2, please.
Okay, yes. So, okay, now the thing is, actually by the time your chromatogram is generated, right, immediately you can do the fractionation. But the thing is, right, usually my advice to my user is you run a trial injection first. Because first of all, right, if you have never run that sample before, right, you have no idea when the compound will be eluted, right? So usually I would suggest to my user, right, you run 1 injection first to know when your compound will be eluted. Then only you, based on that chromatogram, you program your fractionation parameters. Okay, then after that, you save your method, right? In your next second injection, with the fractionation parameters is there, then it will do the fraction collection automatically. Okay, I hope I answer your question, Bernard. Okay, if you need to understand more, you can always contact us. We will be glad to help you. Okay, so my answer to your question is run 1 injection first to know where your compound eluted. Then after that, with the fractionation parameters saved in your method, second injection will be auto fractionation.
All right, so thank you, Wanton. So for our next question, so this is has something to do about our analytical intelligence features. Yeah, so that's something about that we are really proud of because it really increases the productivity in the laboratory. So personally, I have experienced this AI, and I assure you guys that this will significantly help in your analysis workflow. So now the question is, since we are talking about AI, do you have any plans to make an AI program that fully simulates an experiment without using any required physical materials like compound or elements for the experiment? Because in this way, we can mitigate and prevent further harm, yet the program may fail or post different results. So what is your opinion about it? So it's like a Miss Universe question. But anyway, yeah. So Wanton, please answer this question.
Wow, I would say this question is another level already. Thank you, Mark Ivan, for this question. This is a very interesting question. I believe I only received this kind of question from our friends from Philippines.
Yes, we too want to. It's our first time to encounter this question.
Very innovative question.
Correct.
Yeah, very innovative. I like that because this is like idea sharing. Yeah. So, okay, Mark Ivan, I want to tell you that actually in our Shimadzu headquarters, right, we have because in view of the current situation, right, because of this pandemic, now we understand how important is the automation. We all know that now it is so important. I will say that the COVID-19 has accelerated the automation. Okay, because we have no choice but to have more automation in our work life. So actually, in Japan, we have a department that mainly do the automation. So actually, there are more and more automation products in the pipeline that they are designing, they are discussing, and they are innovating now. So Mark Ivan, I'm sure to tell you, because I'm not under the R&D team, but we do have an R&D team for the automation projects. So stay tuned, and then we will have more and more automation products in the pipeline, and it will be launched sooner or later. So please stay tuned with us. Okay, so I hope I answer your question, Mark. Okay, and thank you for that very innovative question from you.
Yeah, so okay. Okay, so for our next question, so I think they have already the Nexera, Shimadzu Nexera system. So can I upgrade my Nexera X2 tandem with LCMS MS in this new system? So I think he is pertaining to our new Nexera series.
Oh, okay. Yeah. So, sorry, can I understand better? Meaning that he wants to couple the triple quad with a new Nexera LC40 series? Am I
correct?
Right?
Yeah, correct.
Oh, okay. Can. So you can. Basically, the X2 is the 30 series. You can upgrade to a 40 series because the Nexera X2 do not have the AI function. So with the new Nexera LC40 series, you have the AI function and you can couple with our triple quad. Okay, so no problem with that. Okay.
All right. So that's just CP says that our system exhibits the high upgradability. Yeah. All right. So I think Wanton, that would be our last question for now. But don't worry, guys, if you still have questions, and for Wanton, for SPC, so you can freely send us a message, send us an email. And we will be glad we will be very glad to accommodate your questions. So for more Shimadzu webinars, you can access those in Shimadzu digital classrooms, wherein Shimadzu now allows you to learn at your own pace, at your own convenience, anytime, anywhere. Why? It is an innovative tool that is packed with Shimadzu webinars, recorded webinars on various analytical technologies, applications, solutions, flow of information, and compliances. And for more information regarding Shimadzu products and services, just visit our official LinkedIn and Facebook pages and feel free to send us a message if you have any inquiry. So we also have a YouTube channel. You may subscribe, please, and watch a number of videos featuring Shimadzu products and applications. Actually, the recording of this webinar, I will be also uploading this in our YouTube channel. Okay? And some of our previous webinars are already there, so you can rewatch it. So this finally marks the end of our session for today. Thank you everyone for participating. Thank you to my SPC colleagues. Thank you, Wantung, for being our key speaker. And we look forward to your usual support in our succeeding webinars and other marketing activities. So thank you again and enjoy the rest of your day. So keep safe, everyone, please. And see you next time.
Take care. Bye-bye.
Excellence in science. Shimazu.